Welcome to Unit 6 of Module 2. In this unit, we're continuing our discovery of the sciences associated, some of the sciences associated with mummification. And in this unit, Unit 6, we're going to look at decomposition and relate that to the process of mummification. If we understand how we're supposed to decompose, we can better understand how we mummify. In other words, how we halt decomposition. And there's a small subunit in this presentation called taphonomy. So we're going to be looking at how the body is impacted upon what things can change and alter the rate of decomposition and maybe alter and even avert mummification. So let's dive right in and first start looking at decomposition. There are generally 5 stages of decomposition. Some references will say 4 and they stop at the post-decay stage, but other references will give you 5, and I like to use 5 because the dry decay kind of gets into a semi-mummified state anyway. So the first stage is known as the initial decay. Something's just dead, and they're starting to, the insects are starting to come in. Then we'll look at these stages in detail. Then there's a bloating stage where there's putrefaction. Gases are starting to be released. Then there's a decay stage. The putrefaction changes to black putrefaction from a green in the bloating stage to black. And then there's the post-decay stage where we get to butyric fermentation, where bones actually start to be exposed. And if it passes butyric fermentation, we actually get into the dry decay stage. Now, we're going to look at a video in a little bit, and you can avert your eyes. If you've been having your kids watch some of these videos with you, you might preview this one before you show it to them. All right, let's look at some of these stages in detail. First stage, initial decay, commences at death and ends with initial bloating. So the individual dies, and the end of the initial decay or fresh stage is when they first start to bloat. And when we look at the video, I want you to try and pick out these stages. Externally, it appears fresh, but there are flesh flies and blowflies, and they begin to lay their eggs. But keep in mind, while this is happening on the outside, internally decomposition has begun. There's cellular lysosomal enzymes which begin digestion, and that's called autolysis. It's a lot of words, cellular lysosomal enzymes. Say that 10 times fast. We're going to look at that in a little bit more detail later on. There's normally present bacteria in our bodies which add to decomposition. They're not held in check by the normal body homeostatic mechanisms. Normal bacteria that we have in our gut, on the surface of our skin, begins to add to the decomposition pattern. And finally, initial decay, because of the cells disassembling and the bacterial action in their own enzymes, leads to liquefaction. So the body begins to liquefy. As it liquefies, gases are released, and that's where we get the initial bloating at the end of the fresh stage. Stage 2, the bloating stage. This is where putrefaction begins to set in. This happens on the 2nd to 6th days post-mortem. And this is when there's digestion by enzymes of cells and bacteria, and they release gases, as I mentioned, into the corpse. And these gases of what give us the swollen appearance. It's the gases that are in the body, but it's also gases that are a result of the decomposition and the enzymatic activity of these bacteria. And it's in stage 2 where you can actually begin to smell the decaying flesh. So it's around the 2nd to 6th days. The bloating stage in putrefaction also first presents with a green flush over the abdomen. It's still stage 2, by the way. So it's over the abdomen bloating, and that's essentially where decomposition starts, in the gut. It spreads to the trunk of the body, the upper part of the body, then the limbs, and then the face. So we get this bloating starting from the belly, essentially, in the abdomen, the pelvic area. And it moves upward, down into the legs, out into the arms, and up into the face. It's not a pretty sight. In fact, bodies can explode during this bloating phase. And the ground zero of bloating, as I mentioned, is a pouch in the intestines called the cecum. Here it is right here, the cecum. And that's where most of the enzymes are, most of the bacteria are that aid with decomposition. The cecum is full of anaerobic, that means they don't need oxygen, bacteria, and now they're unchecked by the normal homeostatic mechanism, so they can start decomposing, taking apart the body. They're there in your bodies right now. They're just held in check by normal homeostatic mechanisms. Ew. Now, these bacterial enzymes decompose the intestinal wall and spread through the circulatory system to other parts of the body. And in the bloating stage, because of the liquid that's now starting to be exposed and liquefaction that's occurring, there's an increased number of blowflies as they continue to lay their eggs. Isn't this wonderful? Let's look on to the third stage, so the decay stage. Now, this is black putrefaction. The flesh now has a creamy consistency with exposed parts that are black in color. So we move from green bloaty to black putrefaction, sort of a black bloaty. Now the gases from the bacterial activity mixes with the blood, and thus the green skin was initially green skin, now it's black by the 5th to 11th day of decomposition. So the second stage was about the 2nd to 6th day, and now in the third stage we have 5th to 11th. And notice there's some overlap. We're going to talk about taphonomy later in this presentation, and that helps us explain why there's an overlap. The film that you're going to see was produced in Papua New Guinea, where it's a very humid environment. So we wanted to see the rate of decomposition there and help us understand how mummies were made in Papua New Guinea in those extreme environments. So those day frames are very important to monitor. The gases inflate the body until pressure increases to the point that body fluids are forced to the outside of the body. The body can actually pop in this phase. I think there was an episode of Bones where the body popped open. Oh God! And goo flew out. So the body deflates after the fluid is forced outside of the body. There's an increased odor, sulfur gases, putrescin and cadaverin are some of the gases, and it's not a happy smell. As body fluids dry up, flies leave, beetles come and feed on the drier tissues because now the body's starting to dry up. And remember, as the flies laid their eggs, the larvae grew, and the maggots grew and began to decompose that body. And stage 4 is called the post-decay stage, or butyric fermentation. It's usually the 10th to 25th days post-mortem, and it's dependent upon the ambient conditions. So here the corpse begins to dry out. The remaining gases percolate, lift the skin away from the muscles, and dermestid beetles comes in. Now these are flesh eaters. These are found in museums and can attack even the dry mummified tissue as well. And then the skin blisters and sloughs off, and finally we have dry skin, cartilage, and bones begin to show. Now, in the video that you're going to see, remember this was conducted in a human environment, and our experiment was to determine, it was using a pig, it was to determine the rate of decomposition in this humid, moist environment. We suspected that it would be faster. In fact, look here, we're at the post-decay stage, butyric fermentation, supposedly 10th to 25th days. We reached butyric fermentation in 7 days, so 7 to 10 days. So it was quite a bit faster than on the faster end, I should say, of the normal stages of decomposition. So let's take a moment and look at that video, then we'll come back in and look at stage 5 and how all this relates to mummification. Here we are on the initial stage. Look at all the flies laying their eggs. You can start to see some decomposition. So this is stage 1. Now we're starting to see some of the maggots, the eggs that have hatched and starting to eat away at some of the tissues. You can't see the initial bloating, but now you see bloating. So we've passed through stage 2, and we've got that black bloating that's going on. Moving through stage 3 and finally to stage 4. Look at the bones there. We're in stage 4, butyric fermentation. All right. We've gone through the first 4 stages of decomposition. I know that was quite a graphic video, but that's how we return, how we decompose, how we return our elements to the earth. There is a 5th stage of decomposition we did not see in the video. We didn't carry on the experiment that long, but it's called dry decay. And this occurs on the 25th plus days post-mortem. So in a forensic case, we're trying to use these stages of decomposition to determine when someone passed away. But we also have to consider what's called taphonomy, the environmental conditions that were there while this individual was decomposing. Just like in the case of our pig in Papua New Guinea, very humid environment, we had to factor that in. We knew that decomposition would probably be more rapid. And it was on the fast end of the normal scales. So dry decay takes place in 25th plus days post-mortem, and mainly bones and hair remain. If you've ever seen a dead animal that's been on the side of the road for many days and has been crushed by cars, you do finally see that the skin, hair, and bones tend to be what remains. And in essence, we do have some potentially mummified tissues. Obviously, this is not how mummies are made because they don't look like little flat things on the side of the road. The corpse is almost dry due to drying out or desiccation, and from this point on, there's a slow rate of continued decay, and the odor is there only slightly. And there's markedly less insect and beetle activity. So keep in mind, even mummies may be decaying slowly as well. Now, there are barriers to decomposition and irregular decomposition. There are physical barriers, the type and nature of the soil. If the person was died and was in the water, if they were in a casket, and the nature of the casket, were they lead? Antemortem and post-mortem injuries. Were there openings in the body that influence the rate at which fluids left the body? There are also chemical barriers. We use embalming agents, insecticides, lime. All of these things can slow down decomposition. Climatic barriers such as heat, cold, wind, rainfall, and humidity can all alter the rate or cause an irregular decomposition. And also there's predation through animals, birds, mice, rodents, dogs, cats, etc. They can all increase the rate of decomposition by coming in and taking a body part and moving it somewhere else. But as you look at this list, looking at the soil, looking at heat, looking at chemistry, embalming agent maybe, or natron, you look at factors here, and what we see are the ways in which mummies are made. Those that have been made intentionally and those that have been made accidentally. So what happens is, of course, decomposition is stopped. And in some mummies, decomposition is stopped early, and we've got a great set of internal organs, lungs, intestines, and everything. And in other cases, decomposition is started a little bit further along, maybe just past the bloating stage. But decomposition is stopped because liquids left the body or the surrounding conditions dried them out. So keep in mind, when we examine a mummy, we examine the external environment as well as the internal environment to determine the state and quality of preservation. Now, we've been mentioning enzymes, and it's important to consider the enzyme trail in decomposition. So we have death. That leads to a cessation of homeostatic mechanisms that keep normal bacteria, particularly in our gut, in check so that they don't eat us up. We have them in us right now. There are proteolytic enzymes that break down the cells because we have no homeostatic mechanism. So the proteolytic enzymes of both the bacteria as well as our own lysosomal characteristics within the cell begin to break us down. The enzymes break down large proteins, fats, and carbohydrates. Larger molecules become smaller and smaller, and it's sometimes enhanced by enzymes from exogenous insects, extrinsic, outside of the body, and molecules are now soluble in water or escape as a gas. And these molecules then join the biochemical pool in water, soils, and atmosphere that is our biosphere. And that's how we disassemble, or we are disassembled after death. We return to the earth in elemental form as gases or water-soluble or molecules themselves. So, what we were is then used by other life. We're part of water, the soils, and the atmosphere. Other life lives off of our elements. It's a little gruesome to think about, but that's how it's done. Now, during the process of tissue softening, liquefaction, and gasification, occurs, and there's a reaction with the environment and living agents such as bacteria, insects, and predators. So as we stated, we have these nice little tight little stages of 1, 2, 3, 4, and finally 5. But there's a reaction with living agents from outside the body, extrinsic characteristics of decomposition. And one of the first things we have to recall is that decomposition happens at the cellular level. It's a Terminator situation when the body dies. Hasta la vista, baby. And here's the cell. And I want you to focus on one of all these little guys inside here. They're called organelles. You probably remember this from bio class, and some of you are probably going, oh boy. Here we are back in bio class, but this lysosome, its main job is to break down molecules that enter the cell so the cell can use it or digest them. You can think of it as the stomach. And in our stomach, as you know, it's full of acid. Our cecum is full of this dangerous bacteria that we keep in check. So the lysosome, in essence, is the stomach, and it is kept in check. All these little yellow things are lysosomes. But as it normally breaks down molecules that enter the cell, at death, their enzymes they aren't held in check. Their enzymes are released, causing what we call autolysis, or self-dissolving or disassembly of the cell. So the cell begins to disintegrate. Enzymes from bacteria, as we mentioned before, further enhance liquefaction. So what was a nice cellular structure now becomes liquid goo. So there are a lot of factors that may inhibit enzyme action, and this is how mummies can be made. Other enzyme action, bacterial action, insect breeding, and feeding all require a liquid environment. So if we eliminate the aqueous environment, we can inhibit enzyme activity. And that's what natron was used for in the ancient Egyptians, to dry out the body, eliminate the aqueous environment. With an inhibition of enzyme action, decomposition can be delayed or halted, leading to desiccation or mummification prior to complete decomposition. And again, somewhere along that continuum. The Egyptians, that we'll talk about them because they had the most well-known method of inhibiting enzymes. They'd take the organs out, they'd make sure the fluids were out of the body using natron, and they'd produce excellent mummies. Now, the aqueous environment, as I mentioned, is required for enzymes to function. So if we remove that water by heat, osmosis, which is what natron uses, and evaporation, all of these can enhance rapid desiccation and ultimately mummification. Heat, inside a crypt or particular climates, just out on the Atacama Desert, Italy, Ireland, Mexico, these are places where mummies are made because heat has a way of removing the fluid, evaporation, enhancing evaporation, and decreasing enzyme function. Decomposition stops. Osmosis. Now, natron is a high salt-like limestone, very basic, so it draws water towards it. So if that's in contact with the body, it draws water out of the body and into the natron or into the limestone, such as in Egypt and in some places in Italy as well. And of course, evaporation. This is exposure. The dry bodies on the cliff sides in the ancient Anasazi region, for example, the air, the dry air ventilating, blowing by those bodies. There's also a wicking effect of mummy wrappings in South America. They put on wool wrappings and fardos, and these draw moisture away. Just set a paper towel out on a counter, and it's the same concept. You put some water there, just set the paper towel there, and that paper towel will absorb much of that water. That's what wicking is all about. So as it's wicked, it's drawn away from the body, it evaporates into the dry environment surrounding the textile, and then more moisture can be drawn out of the body. So it acts like a wick. Also, acidity and alkalinity, particularly in the soil, can impact enzyme activity and inhibit it. So an altered pH environment inhibits enzyme activity. By extension, if we inhibit enzyme activity, we inhibit decomposition. If we inhibit decomposition, we can create a mummy. In acidic environments, we have bog mummies that are produced because of the way that they inhibit enzymes. Plus, they deplete calcium, so other things happen to bog mummies. If it's a very alkaline environment, we get what's called adipocere. The body fats actually turn into, adipose, turns into cere, sort of a waxy-like substance, adipocere. So they're a very unique type of mummy. That can happen in very alkaline waters as well. Temperature is another factor that may inhibit enzyme action. The lower temperatures decrease enzyme action, so cold, dried environments delay decay. For example, the high-altitude mummies of the Andes in northern Italy. One was buried in a glacier, some are buried in a glacier, and some are out there where there is ventilation. As a dry atmosphere, moisture leaves the body. So in other words, the enzyme activity is reduced by the temperature, and the decay is delayed and allows enough time for the body to dry out in these environmental conditions. There's something also called substrate specificity, and that's an altered reaction due to altered structural features of the tissues. Substances like alcohol and formaldehyde denature the protein structure. Remember, the enzymes want to eat protein, but The denaturing of protein results in tissue incompatible with enzyme activity. So there's no decomposition. You've seen back in when we used to use formaldehyde in laboratories, you've seen specimens preserved in formaldehyde. Formaldehyde is one of the main ingredients in our modern embalming fluids as well. In fact, when they first used formaldehyde, they used to use too much, and they actually made mummies. So substrate specificity is another factor that may inhibit enzyme action. Now, certain chemicals can be inhibitors as well. They can slow or halt enzyme action. They may react with the enzyme directly or produce an altered environment, such as many of the heavy metals that we mentioned, zinc, lead, mercury, even nickel, arsenic. And the good examples of these heavy metals, mummies, where enzyme activity has been halted and mummies were made, were Civil War mummies and sideshow or carnival mummies. There are also medical mummies that were made in mercury solutions and arsenic solutions, and we'll be talking about those a little later on in the course. So heavy metals can disrupt enzyme activity. So here's our list. Aqueous environment, if that's reduced, we can inhibit enzyme activity. Acidity and alkalinity in these aberrant hydrogen ion active environments or basic environments, the acid environments and basic environments, we can inhibit enzyme activity. Varied temperatures, both warm and cool, can inhibit enzyme activity. And if the substrate, if the proteins are denatured and altered so that the enzymes cannot decompose them, we can produce a mummy. And then the chemical inhibitors, particularly the heavy metals that we discussed in the last slide. All right, so we now know what decomposition is, and you know from this presentation how mummies are made. Decomposition is stopped by one of those mechanisms that we just described, a reduction in the aqueous environment, the acidity or alkalinity of the soil, and so on. But now there's something that can also impact both mummy production and the rate of decomposition, and it's called taphonomy. So taphonomy technically is a scientific study of fossilization, the living organism's path to fundamental elements, and that's what decomposition is. It's the impact of environmental factors on the process of decomposition. So when we consider a mummy that we find and look at its state of decomposition, we also have to know something about the environment in which it was found. Also, the same is true for a forensic case. Trying to state the time of death based on the state and rate, the stage of decomposition, we have to consider taphonomy. What was going on? What environmental factors were involved? Fauna, animal modification of the remains. Animals can trample the remains, and that can let fluids out and change the rate of decomposition. They can gnaw at them, they can eat them, they can digest them, they can take a body part off, chew it up, and put it somewhere else. And in the soft tissues, there can be pecking and consumption by carrion birds as well. So we have to consider the animal kingdom and what we call predation on the decomposing individual. Also, the environmental impact. Has it rained? What is the climate? What's the average temperature, humidity, wind? We also have to consider gravity. Where has the fluids gone in the body? We call it lividity. It moves towards gravity. So if it's moved towards gravity, there may be more infestation, or there may be rain that's pooled at a certain part of the body, and a certain part of the body decomposes at a different rate than the rest of the body. It's been running water that pulls and draws things away, freezing and thawing, sun, the soil pH as we talked about, and also the topography, which includes, has there been a ventilating environment, if you will. Now, the bioarchaeological application of taphonomy is that we have to consider the temporal context, the time frame from when this person came, the regional environment, the reconstructed environment. That means what was the environment like then when they were alive, the temporal context. They were alive 500 years ago. Was the rain pattern, were the rain patterns the same? We have to learn this from a study of geology of the area. Were there perimortem events? For example, are there slash wounds on the individual? Did they lose fluids? Did that enhance their ability to become a mummy by decreasing the aqueous environment and inhibiting enzyme activity? And we need to distinguish between human-induced versus nature-induced changes in the body. In nature, we're talking about extrinsic factors as well. So in terms of the temporal context in relation to the remains. Was it hot or cold, dry or wet? Was it on the surface? Was it buried? At what depth? Was it in a cave? What was it? A tomb, crypt? Was it perimortem trauma? The state of health, which can influence. If they're dehydrated, they may have a chance of mummifying, for example. Were there any artificial substances in play? Physical composition, age of death, were they clothes, wrapped? All these factor in to the potential for someone to be made into a mummy. Now, it's important to consider these factors because you think back from taphonomy here to taxonomy. If we see mummies, we have to first not assume that they were intentionally made. We might better assume that they were accidentally made and then try and discover if they were intentionally made. Because of all these conditions here, these, depending upon the wet or dry climate, so on, these can influence the way in which decomposition can be halted and mummification can occur. So we have to consider both intrinsic and extrinsic factors. The intrinsic factors are all those stages of decomposition that we talked about. Also, you have extrinsic factors that may influence it. But look at those that relate to the surrounding environment. Has there been human intervention? That's considered an extrinsic factor. Did someone make a mummy? Both intrinsic and extrinsic factors are interrelated and may impact decomposition degree and rate. So we have to try and consider all those, both in a forensic case and when we're looking at mummies. So there's a term in mummification called diagenesis, and that's the changes that take place as a result of increased temperatures and pressures, and this is moving someone towards fossilization. So it occurs, diagenesis occurs faster in soft tissues, slower, the slowest process is in the teeth, and that's why we try and use teeth for like DNA analysis. Bone morphology changes and bone compositions can change. Now that's important, we'll get to that in just a second, as we try and make paleopathological determinations about bodies, and the impact upon bioarchaeological assumptions. So we have to consider intrinsic and extrinsic factors when we're considering the diagenesis of an individual. Now, soft tissue taphonomy. Soft tissue is changed. It is different than bone. So it is, there can be an acceleration of mummification if there's dehydration with heat or chemistry which alters the substrate. Altered tissue states can occur with an altered pH. In bog bodies that were put in peat bogs, they didn't know they were making mummies, but the peat bogs, in order they know they were acidic. But there's also tannin in peat bogs. It's like what's in tea. And that can lead to soft tissue preservation. But another thing happens in the bog we'll talk about in a second, to the bones. While the soft tissue is preserved, the bones dissolve away in a bog mummy. Now, ash is alkaline, and that can lead to, if someone is in a sooty burial site or where there's been a fire, that can lead to adipocere formation because that's alkaline. So the altered tissue states can occur there. Now, bone taphonomies, bone composition, separate from the soft tissue. Now, bone has calcium phosphate and calcium oxide, and it has collagen proteins. So it has calcium phosphate, calcium oxide, and collagen proteins. Well, collagen, let's follow this path first, is reduced through age while we're alive. But after death, the soil pH can disassemble, and higher temperatures can dehydrate and make the bones brittle. So the collagen can look, this is taphonomy. Now, collagen is reduced through age with death while alive, through age while alive. So here what we're talking about is osteoporosis, the loss of density of the bone. But someone, even if they die young, if they're in the right conditions, can look osteoporotic. So we can't necessarily make a judgment about the age of an individual based on osteoporosis, the advancement of osteoporosis, unless we take into account the taphonomic potentials. Higher temperatures, soil pH. Now let's look at the calcium. Calcium is reduced through age while we're alive. We know that, and that's what gives us our bone density. So after death, though, the pH, in an acidic pH, it removes the calcium. So the bog body bones are essentially shells. They're not solid anymore, but it's not osteoporosis. It's a factor of the taphonomy. And water, percolation and soaking decreases the calcium in the bone as well. So we can also have decreased bone density brought on by taphonomy and not advancing age. So it's important to consider taphonomy when we consider the bioarchaeological record that is the mummy that we're talking about. Okay, so we have to consider the impact of varied mummification methods. Were there extrinsic factors, human factors, intentional mummification factors? And does that then disrupt the way in which a body decomposes over 10 years, 50 years, 100 years, 1,000 years, 2,000 years, and so on? Consider thousands of years now of environmental variation. So the point is that interpreting the bones of mummies and the soft tissues is difficult. Because, as I was stating before, osteoporosis, or is it calcium depletion from taphonomic variables? So we have to know a lot about, and as you can see, it brings in other sciences, a lot about the environmental variations this individual is exposed to. Remember, most mummies are still undergoing taphonomic change, even if they're on exhibit in a museum. There are attempts made to keep taphonomy from continuing to decompose or conduct its diagenesis on those mummies. There are special vaults that are made that are devoid of oxygen and so on. But nonetheless, it's a battle for every museum who holds a mummy, and for every mummy that's still in a tomb or underground, they're still undergoing some taphonomic change. So that ends our discussion of Unit 6. We've learned about decomposition through mummification and about taphonomy. And we're going to be moving on into looking into the world of mummies. They're all over the world, as I mentioned. And thank you very much for your kind attention. I think now you have a better understanding of how mummies are made from a scientific point of view and how they may be continuing to decompose even though they don't appear to be so, using the construct of taphonomy. Thank you for your kind attention, and we'll see you in Module 3.